Hello, it's my pleasure to be here, and I'd like to thank the organizers of this committee and Helen for the invitation to speak at this virtual conference. So my role today is to discuss whether precision medicine is the future for cholangiocarcinoma, specifically building on our current treatments. These are my disclosures. So first of all, what are our current treatments? You will know that surgery has been the cornerstone of long-term cure for patients with cholangiocarcinoma, but this is limited to patients whose disease is diagnosed at an early disease. And you've heard earlier about the role of surgery and transplantation for these patients. We also now know that adding chemotherapy following surgery can reduce the chances of cancer coming back. Over the recent years, there have been 3 phase 3 randomized trials which were asking the question about chemotherapy benefit. One was using gemcitabine on its own, one was using a combination of gemcitabine and oxaliplatin, and both of those studies were negative. However, the BILCAP study, which used an oral chemotherapy called capecitabine, showed that by using the capecitabine for a period of 6 months, the risk of death was reduced by 29%. So our standard of care now is to have surgery followed by adjuvant capecitabine chemotherapy if the disease is early. However, most patients present with advanced disease, which is beyond the scope of any surgery. And then we think about systemic options in terms of chemotherapy. This timeline shows you that there has been quite a big gap since the ABC02 study, which validated the use of a combination chemotherapy of cisplatin and gemcitabine, which we showed improved overall survival. Around the same time, colleagues in Japan were also able to show that this also applied in Japanese patients. But despite a number of trials, we haven't really moved forward, and cisplatin and gemcitabine remains the standard of care in first-line treatment. Much more recently, the results of the Advanced Biliary Cancer, or ABC06 study, have reported and just been published in Lancet Oncology. And this study showed that there is a survival benefit from having second-line chemotherapy using a combination of drugs called FOLFOX, or oxaliplatin and 5-FU. With each of these treatments, our improvements are gradual, and we're making incremental improvements, but we still need to have better treatments for all of our patients with advanced disease. So how do we use these in reality? So these are the ESMO guidelines. ESMO is the European Society of Medical Oncology, and they're now 5 years old. But you can see that the first decision point is whether patients do have early stage disease or advanced disease, which could be either locally advanced, so it's wrapped into the blood vessels and operation can't be done, or metastatic, where it's spread to other organs. For patients with early stage disease, we've already talked about surgery, and previously adjuvant chemotherapy was included, although the level of recommendation was quite low because the clinical trial data was just not there to support it. On the back of the BILCAP study, now we have a much stronger level of recommendation, and in fact, adjuvant chemotherapy is now advised for all patients after surgery. You'll have heard from the previous presentation about the role of radiotherapy in patients who have localized disease, and therefore I'm now going to talk about the role of systemic chemotherapy as well as targeted therapies. And just to quickly recap, chemotherapy has been highlighted. I've already spoken about these 2 regimens. But at the time of the publication of these guidelines, the role of targeted therapy, you can see here, there was no standard. But even then, it was beginning to change, and we've learned a lot about molecular targeted therapy in cholangiocarcinoma. So my role is to talk about precision medicine. What is precision medicine? So this is where we take a population of patients. So here, for example, all patients with cholangiocarcinoma. And we can then undertake a process of profiling, molecular profiling, looking at the tumor under a microscope and running special tests, which will look at the genes, the proteins, and sometimes this can be, for example, a blood test. And Professor Bridgewater in the next presentation will talk about this in more detail. But what we're then hoping to do is to categorize patients into groups where we know that there is a specific driver mutation which is driving that particular cancer and for which there is also a specific treatment. So this is now linking the target and the treatment rather than treating everybody the same, hoping that some people will respond and accepting that many will not. So our thinking around cholangiocarcinoma and biliary tract more broadly has been to move from an anatomical classification more to a molecular classification. That doesn't mean that an anatomical classification is not important. You will have heard about the definition of intrahepatic cholangiocarcinoma starting inside the liver, perihilar starting in these ducts just at the exit of the bile ducts from the liver, as well as distal, which are these ducts further down as they go towards the intestine. This anatomical classification is really important, particularly when you come to think about surgery, because surgery is very much based around the anatomy of what's possible. However, when we're talking about systemic therapies, then we can think about the liver more in this type of setup where we've got subgroups of patients who have very discrete mutations or alterations that we can then try and target either through clinical trials or through standard of care treatment. Some of these patient groups are rather small. Some are still to be defined. But what I'm going to do is just focus on two, the FGFR2 and IDH1 to start with, which are the most common alterations at the moment and the most exciting in the field. So let's start with FGFR2 fusions. FGFR stands for fibroblast growth factor receptor. You can see why we call it FGFR. It's much easier to say. So what is FGFR? FGFR is a protein. So this rail here is the outside membrane of a cell. So this is the outside of the cell. This part is the inside of the cell. And what happens is that you have this protein which goes across the membrane. So it's got the outside bit and the inside bit. And in order for this protein to work, it has to twin with another protein which is exactly the same. And a number of different genetic alterations can happen. You can get overcrowding or overexpression. You can see here many more than there should be in that space. Or you can get a mutation that triggers the receptor to work where it shouldn't. And these are marked as little stars here, either on the outside or across both, or even on the inside of the cell. But where this is really relevant to cholangiocarcinoma is that this target twins with another protein that it shouldn't twin with. And the problem is once you get this twinning, you get production of proteins that then drive the development and growth of cholangiocarcinoma. And this is why it is so important to look at what we call these FGFR2 fusions. So what does this mean in reality? Well, this was a clinical trial, a FIGHT 202 study, which is a very important study. Now you're going to see, this is called a waterfall plot. You're going to see a number of these. I'm going to show it a number of times. So you need to think about each one of these bars represents a patient. There is a line here at 0, which means that there is no change in the tumor as a result of treatment. So if there has been a reduction in size of the tumor, the bars go down, so the cancer is better. If the bar goes up, it means that the cancer is worse because the size here of the tumor is larger. Now, within the FIGHT 202 study, patients were treated with an FGFR2 inhibitor called pemigatinib. As you can see, many patients here had an improvement in their cancer. You can see there's a dotted line here at the -30 line. This is what we call a partial response, patients who had an improvement beyond a 30% reduction. Conversely, very few patients' cancers actually got worse. There's a dotted line here at 20. And in fact, the proportion of patients who had disease progression despite the treatment was actually quite small. And between these two lines, we call it stable disease. So the disease control rate is really all of these patients, and that was 82%. So we were able to control the cancer in this particular trial in 4 out of 5 patients. And that lasted for an average of around 7.5 months. Now, this has been a game changer in cholangiocarcinoma. On the 24th of April, so just over a year ago, this was heralded as the first targeted treatment which was approved in the US. And of course, the team here at AMMF were very excited about that, as evidenced by their Twitter feed. But I can also update that this has also now been approved in the EU as of the 29th of March. And so this is one of the treatments that we now increasingly look to find suitable patients for here in the UK. I focused on pemigatinib, but in fact there are many FGFR2 targeted therapies, all with rather strange names, so infigratinib, futibatinib, derazantinib, and so on. And you can see here there's a common theme in all of these waterfall plots. We can see a good proportion of these going down, in other words, the tumor's shrinking, and we can see a very small number where the cancer actually gets worse. So again, a consistent message is coming through that for patients with the FGFR2 fusions, treatment with an inhibitor to target that fusion is likely to at least control, if not reduce, the cancer. Let's move away to a different mutation called IDH1 mutations. IDH1 stands for isocitrate dehydrogenase. This study was different. This was a randomized trial. And in this randomized trial, patients who had received chemotherapy, either 1 or 2 lines of chemotherapy, were then randomly allocated to receive either an inhibitor of IDH1 called ivosidenib or a placebo, a dummy drug. And the randomization was 2 to 1, which means that for every 1 patient receiving a placebo, 2 received ivosidenib. Now, again, we know that this applies to around 10 to 15% of patients with an intrahepatic cholangiocarcinoma. This is called a Kaplan-Meier curve. The higher it is, the better it is. If it's low, that's bad. So as you can see here, patients treated with ivosidenib actually did better than patients treated with placebo. And this is with regards to whether the scan was getting worse or the cancer was getting worse. So in other words, ivosidenib was able to reduce the cancer getting worse by 63%. One of the challenging things of this study is that patients were allowed to cross over. So if the cancer got worse and they were receiving placebo, then they could receive ivosidenib. So that makes it very difficult to interpret what the impact was on overall survival. In a very clean population, you would have patients allocated to placebo who never crossed over. We now know from a recent publication earlier this year that we can correct for the patients who did cross over. And as you can see here, the overall survival data on average showed that if patients received ivosidenib, on average, patients survived close to 11 months. If patients received placebo and then they crossed over, that was 9.7 months. But they can mathematically work out what would have happened to patients had they not crossed over and only received placebo. And you can see here that this has improved from 6 months to 10.8 months. Now, sometimes these numbers appear relatively small, but remember that this is an average. And what any average means is that half of the patients get a greater benefit than this. Some other rarer mutations and aberrations I'm going to focus on now. The next one is called NTRK, or neurotrophic tyrosine receptor kinase, NTRK fusions. This study wasn't specifically done in patients with cholangiocarcinoma. What they did here is they looked for all patients who had this very rare fusion. Again, I'm showing you a waterfall plot here, but in fact, in this study, there were only 2 patients with a cholangiocarcinoma. These are highlighted here, and you can see one here had quite a marked reduction in size of their tumor. In one case, in fact, the cancer got a bit worse. But based on this particular study, the FDA in the United States granted accelerated approval to larotrectinib in patients with solid tumors that have an NTRK gene fusion. And in fact, this is something that can also be accessed here in the UK. But you can see here, this applies to a very small series. 3.5% is probably on the high side. A more recent review from Belgium found less than 1% of patients had this rare NTRK fusion. There's another mutation called BRAF V600E. These are getting more and more difficult to remember, as you can imagine. Again, this is for a very small group of patients. And this was a study called RAW, the Rare Oncology Agnostic Research. And this was, again, specifically looking for patients with biliary tract cancer. You can see here that there's a waterfall plot, which again shows a high percentage of patients who are responding to treatment. 51% if the investigators or the clinicians looked at it, 47% if the scans were looked at completely blindly within the company itself. It took, on average, 9 months for the cancer to get worse, and the average survival, or the median survival, was 14 months. So again, this is another group of patients that we need to be looking for a treatment combined with dabrafenib and trametinib, which again is combining two molecular therapies at the same time to try and get this effect. And the final group to focus on is something called mismatch repair. Now, whenever our cells divide in our bodies, there are likely to be some genetic mistakes or errors that are made, and our body has got an inbuilt mechanism to be able to repair some of those DNA errors. But sometimes that ability to repair is damaged. And in fact, we know that in those groups, immunotherapy is highly effective, because immunotherapy is able to then hone in on the cells that are not repaired and deal with them. This was a clinical trial that included different patient groups, some with bowel cancer, others with different gastrointestinal cancers, and one patient with a cholangiocarcinoma. All patients in this study received immunotherapy with pembrolizumab, and you can see here that the patient with cholangiocarcinoma responded very nicely because they had this mismatch repair deficiency. How common is it? Actually very small, and applies to about 2%, probably smaller in patients with cholangiocarcinoma. Now, COVID has been a challenge over the past year, and one of the reasons why we're meeting virtually. But one good thing has come out of COVID specifically for cholangiocarcinoma. There was a concern when COVID first started about giving patients chemotherapy and whether it increased the risk of them catching COVID or dying from COVID infections. So this nice rapid guideline allowed us to give systemic anti-cancer treatment with immunotherapy instead of chemotherapy to try and reduce that risk of COVID infection and death. And in fact, this was updated in October, because it allowed us to use Nivolumab if we were able to find this mismatch repair deficiency in patients with biliary tract cancer. So COVID has not been bad in all respects. At least it's allowed us to access immunotherapy, but remember, it's for this very small specific group, less than 2%, where they have mismatch repair deficiency. So I've talked about a number of different alterations and mutations, and the importance is that we look, but it's also important that you're aware that it's not possible in all patients. Sometimes the quality tissue sample is not always available, either in terms of volume or quality. And this is a study that we did in terms of our own patients, and you can see here that in just over a quarter of the patients, it wasn't possible to undertake molecular profiling. So in that case, we need to think about either doing a new biopsy or using a different platform like a liquid biopsy, which Professor Bridgewater will talk about next. And even when you do the test, it's important to note that most patients will not have a targeted, potentially targeted mutation that we can give a treatment for. In fact, it's present in around 40% in patients, and most likely if there's an intrahepatic cholangiocarcinoma. So there's still a lot of work to be done in identifying these groups in other patient populations. So my headmaster always said to me, answer the question. I'm going to answer the question. Is precision medicine the future for cholangiocarcinoma? I think the answer is yes, specifically in that testing is increasingly important to see if a targetable mutation is present, either for standard treatments or for clinical trials. But at the same time, it's important to accept that a targetable mutation will be present at the moment in a minority of patients, mostly with intrahepatic cholangiocarcinoma, and it really is dependent on having adequate volume and quality of tissue. Therefore, it is important to continue to improve our standard treatments, as you'll be hearing throughout the rest of this conference. And for the clinicians, the identification of molecular subgroups has also meant that we need to understand much better what the predictive significance is for therapy, the prognostic significance in terms of survival, the impact of prior or post-study therapy, and also we need to understand primary and secondary resistance, because it's likely we're going to want to use a number of these targeted therapies. And with that, I'd like to acknowledge the whole team with whom I work. This was us with our masks on during World Cholangiocarcinoma Day. And I'd like to also acknowledge our funders, Cancer Research UK, AMMF, of course, as well as the Cholangiocarcinoma Foundation and the Christie charity. Thank you very much. I'm open to questions.